This short presentation really is to gear you up to doing research. As you can see from the schedule that was sent to you, there's a lot of the presentations. They're not entirely cancer focused. We've got 2 cancer focused presentations. The others are really related to presentation skills, because you're going to be presenting. We've also got one from Lee Vukovic in Lister Hill Library dealing with researching the literature. And in all your careers, these are going to be important professional development skills. And Lee is fantastic. And she actually wants you to bring in your laptops, and we'll send you a reminder at that time because she's got some exercises for you to do today. So following mine, presentation today will be Dr. Jeff Engler, and he's a professor in biochemistry, but he's also an associate dean in the graduate school. And on UAB campus, he really is the guru for responsible conduct of research. I'll touch briefly upon what responsible conduct of research is, and then he's going to give you much more details about it, all right? But responsible conduct of research is something that everybody has to be very familiar with, all right? So it's very important that you abide by the rules of responsible conduct of research. What are the goals for the summer? All right, although you'll be working on projects that are pre-existing, you're going to be working for a mentor on his or her project. Really what we want you to gain is to learn how to develop an hypothesis-driven, in this case it's a cancer-related or cancer research-focused project, all right? We want you to collect reliable data. We want you to learn how to do this properly. You're going to learn how to analyze your data because you're going to be presenting these data during your actual presentations. And then we want you to learn how to summarize, present, and ultimately, hopefully, you're going to end up publishing these data. All right? And so we're going to have this presentation by a woman in the graduate school, and she's going to talk about how to present your data orally and also how to write an abstract and give you tips on writing skills, etc. All right, so everything that we kind of got outlined follows with what we envision are going to be skills that are going to enhance you as you embark on your careers. All right, so these are our goals, and we hope they're your goals as well. All right, so you all know the scientific method. I mean, you've had this in grade school, right? So it's nothing new to you, but it's important just to refresh what the scientific method entails. So you make some observations, and then you're going to formulate an hypothesis based upon these observations. These observations can be direct, ones that you've actually seen in patients or in the lab or whatever, or they could be based upon a review of the literature. But nevertheless, you're going to formulate an hypothesis. Then you're going to design experiments to test this hypothesis. The experiments themselves really have to test the hypothesis. So when you're formulating an hypothesis, as I'm going to speak about in just a moment, you need to make sure you really can test it. They're going to perform some experiments. You're going to draw some conclusions from these experiments. You may need to revise your hypothesis, all right? Refine it more, and then you can test that new hypothesis or refined hypothesis. And then ultimately what you want to do is you want to publish or present your results. So what is your role as an investigator? Because each of you actually are fulfilling the role of an investigator this summer. So you need to carry out technically competent, right, to the best of your ability. You need to be ethical in the way you do it, and it needs to be thorough, all right? It needs to be reproducible results. Not just one replicate, you need to be able to reproduce your results. Another important thing is, because some of you may have a lot of experience in research, but some of you may have very little experience in research, you need to seek out advice. Seek out advice from your mentors, other people within the lab if you're working the coordinating nurse, fellows, other students, etc. Don't try and run it all along and then you go back to your mentor and say, you know, well, I did this or this or this, but it didn't work. If you're having problems, seek out advice, right? That's very important to seek out advice as you're going along, all right? Then you need to ultimately publish your results. Well, in our case, what's going to happen is you're going to be presenting your results to everyone in this room, all right? But you need to be honest in how you interpret these data, all right? Don't expand upon it if the data aren't there, right? When you make your graphs, make them representative of your actual data. Don't skew the axes in these types of things, all right? People will do that, all right? And the next thing to do is do it in a timely result, or in a fashion, all right? So if you got these results, what you want to do is you don't want to put them on a shelf somewhere, you want to publish them because you spent a lot of effort doing this work. You want the world to know about the exciting things that you've found. So we've got some terms that we're just going to talk about today, all right? Scholarship versus research. What do we mean actually by research versus scholarship? What is translational research? Translational research is a term you hear about all the time. What really does translational research entail? What's an hypothesis? How do you test it? A little bit about record keeping, and Dr. Engler is going to talk more about how to keep accurate records. Process of publication, and then I'm just going to introduce what research ethics, or RCR, really means. So research and scholarship. So when we're talking about research, what we're really doing is we're delving in and we are finding new knowledge. So we're performing experiments and we're enhancing the knowledge. All right, it's brand new knowledge. And typically we're investigating a question or an hypothesis, all right, that's going to lead to new knowledge. In contrast, scholarship, we're looking at pre-existing knowledge. So if you're writing a review article or a Cochrane review, the medical students are probably familiar with the Cochrane reviews. You're going in and you're analyzing lots and lots of pre-published data, and then you're making conclusions based upon that. So that really is scholarship, all right? So that's different between, there is a difference between scholarship and research. So what you guys are embarking upon this summer really is research. And this, in case, is NIH-funded research, so you're falling under the guidelines of NIH when you're performing this research. Translational research. So translational research is really a word that's appeared over the last 10 or 12 years. And everybody talks about translational research. So what do we mean by it? Well, it's biomedical research that attempts to directly correlate basic research to patient care. When it was first formulated by the NIH, it was defined as T0 through T4, going from bench all the way to health policy. You hear people talk about, I'm doing T0, and nobody knew what T0 meant or what T4 meant. It just got more and more confusing, right? Everybody had different terminology, even though NIH had these 5 categories. Well, in 2011, NIH actually decided to formulate a center in Bethesda called NCATS. So that's the National Center for Advancing Translational Science. And this is just their website. And you can look at it because they've got all sorts of really interesting reports about translational research. All right? When they did this in 2011, what they did is they really revised what translational research entails. They got away from T0 all the way through T4. Now we've got basic research all the way through public health research. So everybody this summer really is doing some form of translational research, right? So it's important that if somebody says, are you doing translational research? You can say, yes, I'm doing translational research, right? I'm, doing basic research. I'm doing clinical. I'm doing public health or whatever. All right? So really, NIH-funded research is translational research, right? It's improving public health. All right? So that's the basic differences between these things. Now, here at UAB, there is actually a center funded through NCATS, and it's called the CCTS. So that's the Center for Clinical and Translational Science. And they're over in the Pittman Center on the other side, well, not really the other side of campus, near the hospital, all right? And I put their website up because they've got a lot of really interesting information on their website about courses, lectures. They also, through their Research Common, will help you design studies, perform statistical tests, right? They've got a YouTube channel themselves, and they've got some really interesting presentations on the YouTube channel. So I encourage you to kind of look at this as, as backup material if you're really interested in how research is done here, not only at UAB, but also nationally, all right? So that's the CCTS. So let's look at an hypothesis. Now, this is something you've all had, as I say, probably had in grade school. My wife, she teaches 8th grade science, and they go through the scientific method, hypothesis testing ad nauseum, all right? And the kids at the end of it still don't really know what an hypothesis is, right? I think it's sometimes something we just tend to pay attention to, and then we purge this information, right? But for our cases, we do need to know what an hypothesis is. And you need to know really what the hypothesis is, or the question underlying your mentor's research. Because you want to really understand your research. You just don't want to come in and run the experiments and then go home at the end of the day. You want to really know what's going on with this research, all right? So really, if the mentor hasn't already told you what the underlying question or hypothesis is, you need to ask him or her, what's the hypothesis of this research? All the research, or at least the majority of the work that you're performing, is funded through some kind of grant. Not just the CARES grant, but your mentor has a grant. He or she is going to have to, formulate an hypothesis in that grant. So you need to know what the hypothesis is, right? So what an hypothesis is really just a tentative statement of explanation, all right? It needs to be testable. And that's what gets back to the very first point. When you're writing an hypothesis and designing a set of experiments, the experiments really need to test your hypothesis. So when you're writing your hypothesis out, can you say, can I really test it using my patient population, the lab tools that I have, or whatever, right? Because anybody can generate an hypothesis, but you may never be able to test it, all right? So it's two variables in an hypothesis. There's the independent and the dependent variable, all right? And this differs, of course, from a theory, which is like the global explanation, right? Theory of relativity, theory of evolution. So a research hypothesis is going to state the subject of the study and the expected outcome. So that's the important aspect of it. So when you're writing it, you need to make it simple and clear, right? You need to understand the hypothesis that you're putting down on paper. Readers need to understand, or whoever you're telling it needs to understand the hypothesis. As I've already been harping on, it needs to be testable. You need to formulate your hypothesis in advance. You just don't go and run some experiments and then say, I've got some really interesting data. Let me formulate an hypothesis to match these data. All right? You formulate the hypothesis, then run the experiments. And it needs to be flexible because you need to modify it all along, right? You don't want to go in with preconceived conceptions of this is how the hypothesis is, therefore I only select those data that support my hypothesis. All right? So you need to be really honest in the way you're performing those experiments. And oftentimes you'll find your hypothesis really isn't valid. All right? The experiments don't support your hypothesis or corroborate your hypothesis. So here's an example of an hypothesis on this slide. This is one that, a third grader might write, all right? Temperature may cause leaves to change color, all right? Is it specific? No. Is it testable? No, right? We don't know what the person is talking about with regards to temperature, high temperature, low temperature, leaves, what kind of tree, what kind of plant, what color, right? We're talking about yellow, green, red, blue, purple, whatever, we don't know, all right? So below it is a more specific and testable hypothesis. So it says if changes in maple leaf color are related to temperature, then by exposing a maple tree to low temperature, a change in leaf color from green to red will occur, right? So you can actually go out and test this hypothesis, right? As opposed to the first one, you wouldn't know what to do, okay? So you need to make sure that it is clear, specific, and testable. Now, there's two kinds of hypotheses, right? There are null hypotheses, and then there's the one or two-tailed hypotheses, right? A null one says there's no association between the independent and the dependent variables, all right? A one or two-tailed hypothesis states there is an association between the two variables with or without respect to the directionality of the association, okay? So a null is there's none, and then you can have a one or two-tailed hypothesis. So a one-tailed hypothesis, you're anticipating what the relationship is going to be. So drug A is more efficacious than drug B. Males are more likely to exhibit manual dexterity decline following cocaine abuse than females, all right? So that's one directional. Two-tailed hypotheses can go in either direction, and you don't know the relationship between the two. Drug A is expected to be different from drug B with respect to efficacy. Both males and females differ in response to dexterity tests following cocaine abuse. All right? So when you're formulating your hypothesis, you can formulate a one-tailed or a two-tailed hypothesis. So if we are using statistics to analyze our data, right, we actually have an hypothesis that we're testing. So if your mentor says, oh, you're going to be analyzing your data based upon or using statistics, he or she really has formulated an hypothesis, and you need to find out what that hypothesis is. Now, to achieve statistical significance, right, one-tailed, you don't have to have as large a sample size as for a two-tailed. So the more samples you have, the more rigorous the testing is going to be. So a two-tailed hypothesis is going to be more rigorous than a one-tailed hypothesis. All right? So the point of this is to get you thinking about what the hypothesis is that you're actually working on this summer, right? And how the hypothesis is being tested by the data that you are collecting, and how actually you're going to analyze these data. And so I come back to this slide because Dr. Desmond here, she's an expert in biostatistics, right? But for the rest of us, I don't have any statistical expertise by any means. So what I would do is I would go to the CCTS, and they have actually weekly sessions in which you can talk about how to analyze data, how to design studies, etc. All right, and they have weekly hours, and they're over in the PCAMS building, and you can set up times, and I'm sure Dr. Desmond is equally happy to talk to you about how to analyze data or whatever, right? But sometimes, you get involved in getting lots and lots of really interesting data, and your mentor becomes stumped because the limits of how you would analyze those data exceed the knowledge of that mentor, like it would exceed my knowledge. All right? Maybe you guys are experts. I know the MPH and the MSPH students, they know statistics, but some of the medical students probably aren't up to speed as much on their biostatistics, right? So there are lots of resources on campus to help analyze your data. So the next thing comes to record keeping, and this is something that Dr. Engler is going to talk about to you, but in more detail. But record keeping is very important, all right? Now, the NIH say after a grant expires, you really only need to keep your data for 3 years, all right? And the NSF, it's 3 years past the final project report, all right? However, if ORI, which is the Office for Research Integrity, comes knocking on the door of your mentor saying that, you know, we have concerns, or there's people expressing concerns about the validity of your data, right? And you've been funded by NIH. We want to look at your data. Now, typically it goes through the institution first before it goes to NIH, but regardless, ORI says you have to have 6 years, right? They can go back after the end of that progress, right? So the point being is when you're taking your data or recording your data, you need to record your data accurately, all right, with the thought that these data are going to be retained by your mentor for a relatively long time period. You're going to be long gone, right? You'll be off in your residency, you'll have a job, you'll be doing something else. And your mentor's not going to know who did the experiments or whatever if you don't document it well. So you need to record your data very, very well, just in case something happens, all right? So that's important, that you record your data and you retain your data. So if you're using an electronic notebook, just storing your data on a computer, right? Make sure that your data is backed up. Talk to your mentor about how he or she wants your data backed up. In the School of Medicine, I direct the scholarly activity, and invariably at the time that it comes for 4th year to students to write up their final reports, 1 or 2 each year will come and say, well, I went back to the lab to get my data off that computer, and that computer no longer exists. Now what do I do? Or that computer, you know, was really run by such and such a clinical coordinator. She's gone, right? I don't know how to access. My mentor didn't have any idea where the data was, and so now I'm up the creek, all right? So back up your data, right? If it's patient-based data or whatever, back it up on an encrypted thumb drive, right? But back up your data. But discuss with your mentor first, but you want to have these data, all right? Just don't leave it in the lab or on the clinical computer or wherever. Back up your data. Things are a little bit different in England. They require you to have 10 years for lab-based data, and for clinical data, they want you to retain the data for 20 years, all right? So the point being is that you can help in the retention of these data by backing it up yourselves. The data is not lost. So when you look at a notebook, right, what do you want to record in your notebook, all right? So if you're going to be working in the lab, and some of you are, right, you want to have the name of the experimenter, which will be yourself, the date of the experiment, what reagents that you're using, where do these reagents come from, lot numbers, all these things can vary, right, and impact the results. It's not uncommon if you use antibodies in your work for antibodies to vary between lots, all right? Certain chemicals can be contaminated. You need to record all this. If you're using mice strains or yeast or whatever, record all that information. All your methods, just don't write, you know, if you're doing Western blotting or whatever, I ran a gel and did a Western blot using the following techniques, as usual, right? Detail those. Put down your results, even those that didn't work, right? Just don't discard the bad data and only keep the good data, all right? Put all that down. How you interpret those results, any conclusions, what are your next set of experiments going to be? right? And were these based upon previous experiments? Are these a replicate of something that you may have done a month ago or whatever? Because somebody else may be looking at that data to try and pull the information out for publication, and you're back in courses, you're in clinical rotations or whatever, and it's a lot easier if they can go to a great notebook and find this information. So electronic ones are good, right? Because they're accessible by multiple people. They need to be backed up. You can put multimedia files in them, right? You can have images imported in there, etc. You can have the images imported or information imported directly from instruments, and they can hold an enormous amount of data. So more and more people are going to electronic notebooks as opposed to handwritten notebooks for data. But the most important thing is you record accurately and you back this information up, or make sure that it's backed up. So just as we're running shorter on time here, let's briefly talk about publication, because as I said, we're going to have a session dealing with writing an abstract and presenting your data, all right? Publication is, in legal terms, is the first time a work has been publicly distributed by a defined means, all right? So, I mean, that can vary by what a defined means really means, all right? But if you're presenting it at a meeting and you submit an abstract and that abstract gets published, right, that's a publication, right? Any kind of reports, say you're working on a public health project and it's funded through the State Department of Public Health, but it's more of a white paper or whatever. It gets sent to the State Department of Public Health. It's going to be published in their annals. And that's a published document. It need not be put into a journal format, all right? When you submit grants, and oftentimes when you see these investigations of misuse of data, the NIH are going back and looking at people's grants that have been submitted 10 years or more ago, and they find that figures had been doctored or whatever. So they're part of the literature in that way as well, all right? So what's the point of writing this, your data up, right, or preparing for a presentation? It really allows you to think clearly, all right? Because now you're really going in and investigating what all these data that you've collected really are leading to, all right? So it really does help you clarify your project. You want to actually share your interest in data with the community. For personal gains for students, it's great to have publications on your CV. I mean, that's fantastic. So you get personal gain that way, right? You get to establish yourself as an expert for the MPH students or the MSPH students that are writing theses. You're the expert in that field at that time or on that project, right? And you really want to show people what you've done, all right? So, as I've said, scientific writing can take all different formats. It can be from review articles to abstracts, teaching materials, etc., all the way down to grants, all right? So use the same ethics when you're dealing with any kind of scientific writing, all right? So to be an effective writer, you need to base it on the reader. You don't have it full of jargon. You want everybody to be able to read what you're doing. So when you guys are going to be putting together abstracts for the Cancer Center retreat or Medical Student Research Day or Graduate Student Research Day or whatever, right? You want other people outside of your field to be able to read that abstract and understand what you're talking about. It shouldn't be only you and your mentor understand or somebody else in the lab what you're talking about, right? But oftentimes you'll see that. You don't have a clue what the person is talking about, all right? So you need to be reader-based. You need to be purposeful, you know, accurate. Accuracy is very important, right? Clear, concise. You need to be precise but you don't want to have it going on, rambling on, so it needs to be simple, and you don't have lots and lots of jargon. If you do, you need to define these terms as you write the abstracts. So this is the same kind of process that you guys will be going through, except you're not submitting it for a publication, but you complete your research, preparation of a manuscript. Well, in your case, it'd be preparation of the presentation, right? You're going to be presenting it. In our case, we're going to be reviewing it because you're going to get feedback on how you are presenting, and not on the quality of your research, but really how you are presenting it, right? Then a decision, if it was a real manuscript, a decision would be made. And it could say we're going to accept that manuscript and we're going to publish it. It could be rejected, which means a lot of things, right? You may say, okay, I'm going to submit it to another journal. This was too high tier for me. All right, and when Lee Vukovich comes in, she's going to be talking about, the qualities of journals and things like that when you're really researching things, all right? But it could be rejected, and you say, well, I'm going to change the journal format, I'm going to submit it to a different journal, I need to do more experiments and resubmit it or whatever. All right? Sometimes the journal will say, yes, we really are interested in your work, but we've got these concerns about it. You correct it, we'll send it back out for review again, but we really want you to submit this work to our journal. And then it ultimately ends up in being published again. All right? But it's the same kind of process. By writing an abstract or presenting a report, as you're going to be doing it here in this class, it goes through the same mechanisms. All right? We're just starting smaller than making you write a manuscript. So here's just a couple of books that are good on effective writing. There's one by Michael Alley, and then there's Jean-Luc Lebrun, and they both are good books. You can get them through Amazon if you're interested in how to write clearly as a scientist. The last thing is research ethics, and this is also known as the responsible conduct of research, or RCR, and that's what Dr. Engler is going to talk about. So in broad perspective, it's talking about the ethical and moral principles to issues in scientific research. And these are something you really do have to bear in mind. Design and implementation of research involving human subjects and animal subjects. We're not going to have a presentation on animal subjects, but Carrie Oliver from IRB is going to come in and tell you about the IRB here at UAB. Scientific conduct, how you conduct yourself with the experiments and things like that. There's another issue called whistleblowing. Now, hopefully we don't have any of that. All right? Right? Need. But you do need to be wary of that. All right? You may be involved in a research project, and your mentor's not doing anything wrong, and your mentor knows nothing about it. All right? But you see another student with whom you are working, right? And the data they're getting, just doesn't make any sense. They're hardly ever around, and they've got all these fantastic data. And you really don't believe that this person is doing it correctly, all right? What you need to do is you just don't say, oh, I don't see anything, right? Yeah, you need to, if you're comfortable, talk to your mentor about this. All right? And that's essentially what whistleblowing is. All right? Now, as I say, the chances are you ever encountering this are highly unlikely. But there is a case. There was a case at Duke. There was a man called Anil Patti, and it was on, 60 Minutes, the whole bit. And he was a physician, and he was really big in cancer pharmacogenomics. And he actually, nobody could understand the data, but everybody thought this was absolutely fantastic. And he was going with his mentor, right? They were going and they were putting in patents, and they were going to become billionaires potentially from this. Well, there was a medical student in that lab, and that medical student was on a year out. He had a Howard Hughes fellowship. And that medical student was, he was trying to understand the work that was being done. and he just couldn't understand the numbers and the statistics. It was a lot of, genomic analysis, and he just couldn't figure it out. It didn't make any sense to him. When he ran the data, it just wasn't coming out. So he had a big dilemma, right? What am I going to do? I really think there's something amiss. If I speak up, here I'm on a Howard Hughes Fellowship to work in this lab. I'm a third-year medical student. What do? Do I say, I think something is wrong? Or do I keep quiet, I'm just the naive student or whatever? Well, this fellow, he actually went forth with it, right? And so that other people at other institutions were beginning to wonder. MD Anderson, there were statisticians there, they were beginning to wonder about this as well. But this guy actually had access to the real data. And he went forward, right? And the deans at the medical school were not happy with this, because here was these, these big wig scientists, potential with, millions of dollars coming into the institution. But ultimately what happened is that, he stuck to his guns, and the data that he was able to provide led to an investigation, and everybody that was part of that group are no longer at Duke, and they're really no longer in science. All right? So it's something that this poor student never would have dreamt that he was getting entailed in. All right? But it can happen, and it can happen to a student. All right? I'm not telling you what to do, but this is something that you just never think will happen, but it's possible that it can happen. So just be, wary about that. If you see students or other ones that you just don't quite understand this, go talk to your mentor about this. All right? Conflict of interest is another big thing that falls under this, particularly, if you're getting funded to do a drug study by the drug company and these kinds of things. And it also entails research regulations. There's a lot of research regulations that you need to follow, and that all falls under research ethics. So, to summarize, we've talked briefly about research and scholarship, what translational research is, and I think all of you actually are doing some form of research that falls under translational research. Hypothesis generation, record keeping. It's very important that you record your data well, all right, and accurately, and include the negative data in there as well. Don't edit it, include that negative data as well. Publication, and then research ethics, which Dr. Engler is going to talk about.